nutrient budgets, pools and processes in wetlands. What I wanted to do is just to put that into the context of the connectivity between water quality and land management practices and the reef. Conceptual diagram that I think Ryan presented as well, and it's just showing the connectivity between what's happening on the land, whether that be agriculture or whether that be grazing, and the water quality, which is obviously connecting out to the reef. One of the issues which has become clear in the last little while is the importance of sediment on reef water quality, and more so, not just so much the quantity of the sediment, but the quality of that sediment and sediment size, because there's some good evidence to suggest that while some of the heavier sediments, maybe greater than 63 microns, for instance, get deposited close into shore, the very fine clay sediment, maybe less than 10 microns, gets into the reef system and can be transported very long distances and may be contributing to the COTS outbreaks that we have, because there's this thing that's called marine snow. And the marine snow is this amorphous type of gel, and it's made up of a whole heap of biological organisms, microorganisms, etc. It's also got, as a matrix, very fine clay sediment. And that seems to be the matrix which is moving a lot of the nutrients off into the further parts of the reef than we would have originally assumed if we were just concerned with this heavier sediment which gets dropped on the near inshore. A way of trying to bring it all together in terms of all the work that's being done in the catchments. So in the catchments we've got work being done at the block scale in the cane industry, at the property scale with the grazing industry, then we've got the water quality monitoring network, we've got a whole lot of catchment scale work going on. It's trying to link all those things together to get this connectivity between what's happening on the land and what's happening on the reef. The bit that I've been dealing with in the agricultural context is this property planning and source control. So when we look at a whole pollutant treatment train, we go through the source control, the transport, we can do some treatment here on the property scale, we then have the transport again, we can do some treatment here on the catchment scale, and this is where the wetlands tends to come in. Then we end up with the discharge out into the reef area. The bit I'd like to talk about initially is this part up here, which is the property scale and attempting to control the movement of pollutants off the property. The nitrogen cycle in agricultural systems is very complex, and there's lots of different pathways, and as Ryan said, it can be a very leaky system in terms of where nitrogen goes. Nitrogen's very mobile in the environment. When the growers put fertiliser on, there can be volatilisation losses of ammonia to the atmosphere, but we've got this whole cycle here going on in the soil. We've got the organic nitrogen that's either been contributed to by some of the property management that we might have been doing in terms of meal mud, legume break crops. They contribute organic nitrogen into that pool. We also have the soil organic matter. So we have this soil organic nitrogen, and then it goes through this process, this biological process, from ammonium through nitrate, through nitrite to nitrate. And at any of those processes, when nitrogen is in the ammonium form, the cane plant can take it up. When nitrogen is in the nitrate form, the cane plant can take it up. But once the nitrogen gets into this nitrate form, that's where it's very, very environmentally interesting in terms of, in a leaky system, that's a leaky system. When we've got nitrate here, which is moving by one of these pathways rather than being captured by the crop that we're trying to grow. Under anoxic conditions, we can get denitrification. That's contributing nitrous oxide as a greenhouse gas, so that's a problem from global warming perspective. Nitrate can also be leached by drainage, depending on soil type, and Ryan's or Moody mentioned some of the elevated levels of nitrate in groundwater. And if we've got a situation where there's runoff and there's sediment and erosion, then the nitrate can be moving off the block by that pathway. So in terms of trying to treat the system, in terms of property scale management to try to reduce the leakiness of the system, it's all about the source control, and we're trying to get as much of this applied nitrogen much of this available nitrogen into the crop as we can, because once it's in the crop, it's not an environmental issue. If it's not in the crop and it's in the nitrate form, it may very well be an environmental issue. So that's the situation, I guess, at the property scale. So now let's move down to treatment at the catchment scale, and this is where the wetlands comes in. I'm going to move out of my comfort zone in agricultural cropping systems and step into the mud of the wetlands. And let's have a look at some of the situation or the inflow, outflow of nutrients through a typical wetlands. We can look at sort of what I call a black box nutrient budget. If we're trying to determine whether or not the wetlands are acting as a sink or source of nutrients, then if we take this black box nutrient budget approach, we need to have an inventory. We need to have an inventory of the nutrients which are going into the system and an inventory of the nutrients which are coming out of the system. And as we know with our nutrients, we've got carbon, which is driving the whole nitrogen and phosphorus cycles. We've got the nitrogen cycle and we've got the phosphorus cycle. So characteristically in water quality monitoring, we measure things like dissolved inorganic nitrogen, dissolved organic nitrogen, particulate nitrogen, dissolved reactive phosphorus, and particulate phosphorus. So we can measure those parameters as they go into the wetlands, and we can measure those parameters as they come out the other end, and determine whether there's actually been a change. And quite often, I suspect, we will find that certainly some of these may have decreased if the wetlands is acting as a sink. Some of these may increase if the wetlands are acting as a nutrient source, but it's the changes between those different forms which is becoming hugely important from an environmental perspective. The nutrient processes and transformations which are occurring between those forms which are going into the wetlands and the forms which are coming out of the wetlands governs the effect of those nutrients once they've left the wetlands on the larger environment. Wetlands are, of course, extremely complex. And like our agricultural soils, there's microsites in the wetlands which have got vast changes in conditions in terms of pH, redox potential, carbon availability, biological activity. So you cannot characterise a wetland as being aerobic or anaerobic. You can't characterise a wetland as being mainly this pH or that EC. Not possible, because there's microsites all through the wetland which are all behaving like their own little mini environments. And the complex nature of it is such that if we just measure what's going in and what comes out the other end, well, that'll give us a bit of an idea of an inventory of what the wetland's doing. But it's those nutrient processes and pools in the wetland which are governing what happens in terms of the exit nutrients. The nitrogen cycle in the wetlands is extremely similar to what it is in a terrestrial environment. So we've got those conversions of organic nitrogen through the mineral forms of nitrogen. Once it ends up as nitrate, that's when we can have denitrification occurring, or we can have the nitrate moving off site. So the nitrogen cycle mainly driven by biology, but very heavily dependent on the carbon cycle and on the dissolved organic carbon levels in that environment. On the other hand, the phosphorus cycle is more governed by chemistry. Surface chemistry is what governs the phosphorus cycle, both in agriculture systems and also in wetlands. If we have a look at those two systems, we can see then that in terms of the wetlands, the very important one that we need to be having a much closer look at is the fate of this particulate nitrogen. Particulate nitrogen is just measured in our water quality network as the total Kjeldahl nitrogen in the dirty water with the sediment in it, and you subtract from that the nitrogen in the dissolved inorganic nitrogen, and that gives you the particulate nitrogen. But that form of nitrogen is forming these pools of nitrogen as it moves through the wetland system. It's producing dissolved inorganic nitrogen and dissolved organic nitrogen simply through the processes, the biological processes which are occurring. Phosphorus, same deal. We've got particulate phosphorus And currently our methods don't distinguish between, they measure particular phosphorus as total phosphorus. So the total phosphorus in the sediment. But the total phosphorus in the sediment comprises the mineral phosphorus and also adsorbed phosphorus. And again, a difference between the bioavailability of that particular phosphorus is hugely important in terms of what's coming out of the wetlands. You can see that the quality of the graphics has decreased by about 3 orders of magnitude. But if we're starting to talk about particles, and we're really important part of the particles is their size, how are they going to behave in the wetlands under different conditions? If we have a base flow condition going through the wetlands and we've got some rather large sediment particles coming in, there's a good chance that they might settle on the bottom of the wetland and become part of the bottom sediment. Under a high flow event during a cyclone or a flood, good chance those large particles might be able to bounce through the wetland and pop out the other end. But it's those clay-sized particles, the ones that are less than 10 microns, which I suspect are having a major impact on Great Barrier Reef water quality, and something which in the past we've tended to ignore or not even acknowledge the importance of. And it's these particles which in a low flow event, if there's not enough detention time in the wetland, there's a good chance they'll come in and they'll go out the other end. And during a flood event, you can guarantee they'll just be express trip straight through the wetlands. And it's the impact of these off-site which I think is hugely important. On those particles then, let's have a look at some particles moving out of a wetland or even moving into the wetland. What are the important things which are impacting the nitrogen bioavailability of those sediments? First up, it's specific surface area. The smaller the sediment particle, the greater the tendency for it to mineralise nitrogen, just because there's more surface area there, there's more available substrate for the microbes to start mineralising. Redox potential, a hugely important effect, because mineralisation, the mineralisation process where organic nitrogen gets converted to nitrate, needs an oxic environment. The other side of that is the denitrification process. And denitrification occurs in anoxic environments. The balance between net nitrogen mineralisation or net nitrogen denitrification really depends on the redox potential of the water column or the microenvironment that sediment is actually sitting in. Carbon to nitrogen ratio, also hugely important. If you have a large carbon to nitrogen ratio, probably something greater than 20 to 1 for instance, you end up with what we call net nitrogen fixation. There's not enough nitrogen being produced to be able to be taken up by our crops or microphytes or whatever, because the bacteria, as they're chewing into the carbon, oxidising the carbon, they need that nitrogen for their metabolic processes, so they tie it up. So you end up with a net nitrogen immobilisation for a period of time if you have a high carbon to nitrogen ratio. On the other hand, if you have a carbon to nitrogen ratio of maybe 15 or less, you will get net nitrogen mineralisation under aerobic conditions. So we can think of the particulate nitrogen and carbon as following these sorts of processes. And we've got a time scale here because time is hugely important. The conversion of organic nitrogen under oxic conditions to nitrate takes a matter of hours. In agricultural soils, we reckon if you put on an ammonium-based fertiliser today and everything's fine, you've got a moist soil, but an aerobic soil, within 2 to 4 days, most of that ammonium will probably have ended up as nitrate. So this particular process here, under oxic conditions, very fast. And we've actually we've simulated some sediments. We've taken some surface soil, suspended it in water, allowed it to settle with Stokes' law so that we've been able to recover the less than 10 micron fraction from that soil. And then we've taken those 10 micron fraction soil sediments and we've incubated them for a period of 3 and 7 days to see how much mineral nitrogen gets produced. In some of those sediments, they produced about 5 milligrams of nitrogen per kilogram of sediment per day. This process up here is occurring all the time as that sediment is in the water column. So as that sediment moves through into the wetland and out of the wetland, out to the reef, it's mineralising more ammonium and nitrate as it goes. Under anaerobic conditions, anoxic conditions though, we have the denitrification process that kicks in. And so once we've got our nitrate, under these anoxic conditions, it then gets denitrified as nitrous oxide or perhaps even nitrogen gas. And carbon, same way, as it's oxidised under oxic conditions, it'll go to CO2, but under anoxic conditions, it'll form methane. But that whole process is occurring as these sediments are moving through the environment. With respect to phosphorus, as I said, it's mainly a chemical type situation, but again, the same factors are very, very important. Specific surface area, again, any soil chemical reaction which is dependent on absorption, desorption, the greater the surface area that's available for that to occur on, the greater the importance of that. Redox potential, very important where we've got phosphorus being tied up with iron type compounds. In soils, the major absorption sites for phosphorus are aluminium oxides, hydroxides, and iron oxides and hydroxides. If phosphorus associates with the aluminium and the redox conditions change, then there's no net effect much on the absorption, desorption properties. But if the phosphorus is absorbed on iron compounds and you end up with a redox potential going down, what happens is the iron gets reduced from iron 3 to iron 2, the iron 2 becomes soluble, and the phosphorus which the iron 3 was holding gets released into the water column. So when you go into an anoxic condition where the adsorbed phosphorus is associated with iron compounds, you can have a major release of iron and phosphorus, dissolved phosphorus, into the water column. And the clay mineralogy is what really drives that process. If we've got clay minerals such as the iron and aluminium oxides or the kaolinite type minerals, the 1,1 type clay minerals, they have what we call surface charge chemistry. They're variable charge. And they behave differently to the clay minerals which we have in our heavy cracking clay soils like our black earths and our vertisols. The clay mineralogy of the soil in the wetland, the clay mineralogy of the source soil of the sediments coming into the system is hugely important when we're trying to understand phosphorus bioavailability. So the phosphorus cycle with the particulate P, as I said, dominated by chemistry. Under anoxic conditions, if the adsorbed phosphorus has been associated with iron compounds, then it can certainly become released into a dissolved form. We have this temporal trend occurring again. These reactions, chemical reactions, are very, very fast, but we do have an interesting one with phosphorus because when we add phosphorus fertilisers to soils, a lot of the phosphorus gets absorbed on the soil, but over time it actually moves into the soil particle and becomes less available for plants and for microbes. So we have that slow fixation process occurring. That occurs over weeks or years in the agricultural context, but in terms of these soil P on a soil particle or a sediment particle getting into a water column and going through wetlands, once these conditions occur, those chemistry reactions are very, very fast. We have the situation with the phosphorus attached to a sediment moving through a wetlands or moving through a water column out to the reef. It's those basic chemical properties, absorption properties of the sediment which determine the bioavailability of that phosphorus. If that phosphorus can be easily released, then it becomes bioavailable. If that phosphorus is tightly held by the sediment particle, then in terms of biology, it's not an event. The basic question, I guess, is do the wetlands measure up to a treatment at the catchment scale? And so a basic approach might be, again, going back to this black box nutrient budget. We need to know the nutrient forms going in and the types of forms going into the wetlands. Are there any changes in the levels of those nutrient forms? If there are changes, why? What has occurred in the wetland which has caused the nutrient to transform from one form to another form? Total suspended solids in, total suspended solids out. Has there been any change in the total suspended solids? But total suspended solids are a very blunt tool for telling us about sediment bioavailability and reactivity. We need to know what the distribution of particle sizes are in that total suspended solid load, because it's those very fine clay fractions to less than 10 microns which are going to move a long way in the marine environment compared to the much heavier ones which will probably be dropped in the near shore area. Thanks for your attention.